Good morning. It's a pleasure to be here. Thank you for the invitation to represent the All of Us research program. Many of you may remember this initially being launched by former President Obama in 2015, originally called the Precision Medicine Initiative, and we renamed it in 2016 after realizing that participants don't understand what the word cohort means and doesn't ring as positively as something like as inclusive as All of Us represents. I received my funding primarily from a number of nonprofits. Since starting this, Vanderbilt has created a company that does pay a small license fee for use of its Vanderbilt's resources to Vanderbilt. So All of Us protocol was designed to enroll a diverse population from across the United States and really allow anyone across the country to be able to participate in some manner. So based on this and a lot of our research and use of healthcare systems, we have two modes of enrollment. One is the healthcare provider organizations, which is a more traditional route of using large healthcare systems that have established relationship with patients to bring their patients in, to consent them and bring them and have them join the program, and also gives that facility that they know and trust to enroll and do the physical exam, collect biospecimens. The other route is through something we call direct volunteers, which allows essentially anyone anywhere to be able to enroll primarily digitally, and then we have a number of other providers like convenience clinics like Walgreens and Quest, which is a local lab testing company that has facilities across the country to engage those participants to collect biospecimens. We're also looking at things like salivary pilots to send to remote areas. Essentially, despite how you enroll, it's the same process, the same consent, the same sets of variables that we're trying to capture, which include surveys as well as in-person physical measures, biospecimens, blood and urine, and smartphone data. The goal is at least 1 million diverse participants. We launched nationally in May of 2018. We currently have about 160,000 core participants, which have agreed to share the electronic health records and contributed biospecimens. Over 200,000 have consented. We enroll over 300 sites. We're very excited that we are over 50% non-white and actually over 80% underrepresented in biomedical research, which includes both race, ethnicity, as well as being lower socioeconomic status, sexual gender minorities, and being rural locations, and a few other measures. We have over 100,000 electronic health records that we've collected. Our plan is to start both GWAS and whole genome sequencing, hopefully before the end of 2019. And a key aspect of our program is participants do get data back. This just gives you some screenshots of the portal, and we also have Android and iPhone apps that participants can log in through and complete their surveys and share things like Fitbit data. That's currently the only device that we're capturing data for, but we're expanding and plan to go towards Apple Health Kit and Android as well, and plan to actually do pilots with specific devices also. Capturing electronic health records in the United States with a highly fragmented healthcare system is a challenge. And we have to have, for instance, legal agreements with every entity that's providing data. And so negotiating those, all those legal agreements has actually been one barrier. Thinking about the different state laws that come on top of the federal laws is something we've had to think about as well. Right now we have 32 different healthcare entities representing probably about 50 or 60 different actual institutions sharing data. It's across about 14 or so different electronic health records. Each site is mapping to a common data model, which I'd be happy to talk about if we desire to go there in the conversation. This represents really the first US-based aggregation of electronic health record data from across a number of different entities. And we are finding that so far that is, we've been able to accomplish those barriers and actually share, including things like the full narrative content. We're heading towards that. We're just starting on the narrative content, but that is the direction we're going and have not seen pushback from sites. Our data structure involves, if you think about us kind of representing everything in the middle and up, we have a number of different inputs into a raw data repository, and that includes the participant portal, the biobank, which is at Mayo in Rochester, Minnesota, and then different input collection facilities that we have developed. And then we curate that data, which includes removal of obvious identifiers. We have a couple different tiers of data, which I'll go over, and then that feeds cloud-based research environments that researchers will use to come in and use the data. We have the first of these live now, which is on this Research Hub website, which includes a data browser to explore the data, a way to look at our surveys, download those in English and Spanish, and kind of snapshots of where we are. And then we're working on building that researcher workbench where people will come in and actually use the data for analysis on a kind of row-level data. This is just a screenshot of the data browser, which you can search through our electronic health record data, the physical measurements and surveys that we have. And thinking about when someone comes in to do analysis, we've actually established 3 data tiers, 2 of which have individual row-level data and requires authorization. Our goal is to make this accessible to a worldwide cadre of researchers and really support a broad range of researchers as well. And so as we're starting with the NIH-based ERA Commons ID, which is an international ID. About 300,000 individuals have one now. And then once with a valid ID, we intend to extend beyond that over time. There's a series of training modules that one goes through, and then they agree to a data use agreement. On the registered tier, which is more de-identified. De-identified is actually a formal, has legal meaning attached to health data in the United States, but essentially we all know what that means. It is more de-identified, has more identifiers removed and more towards anonymity on the registered tier. And then on the controlled tier, that's where you'll have things like your clinical notes. Certainly the rarest genetic variants will be over there, at least at the beginning. And that has the additional step of an institutional sign-off and validation of who the individual is. And once you go through those steps, essentially you will have a passport to use the research, the resource, however you want to use it for any allowable project as defined by the videos you've gone through, the training modules, and the data use agreement you signed. Those can be audited and they will be made public as well as part of actually the 21st Century Cures Law, which provides a lot of our funding. This just gives you some screenshots of our beta platform that allows you to create different projects. There's graphical tools to explore the data and create cohorts and to pick which variables you want to put into your notebook. And then right now it starts with notebook-based analysis in Python and R, and we intend to extend that over time as well. So some of our challenges, I mentioned earlier, state laws, they vary for how both genomic information is returned, but also things like how long a EHR consent can last. So we had to modularize that component of it and do some slight variations for different state law. Collecting EHR data from a number of different sites and harmonizing it has been a huge challenge. Thinking about how we balance broad access with security and flexibility of the resources in a common cloud-based environment that we can audit use of. We all know the power and need for analyzing data across many cohorts, and those kind of bridges are something that we are thinking about as we develop this resource. We have picked a common data model instead of vocabularies. As we think about not just in all of us, which I think we have taken for instance, our surveys and map them into the same vocabularies and identifiers that are used in electronic health record data, so that they are at least adjacent to or in a common data architecture. As we think about extending that to other resources, that will be something worldwide that I think we have to think about. Thinking about data quality across different providers and batch effects, not just in genetics, but in phenotyping, and how you minimize those, both on the data providers as well as the data collectors. And then, how we really support broad groups of researchers. I kind of alluded to this. We want to go beyond just traditional academic researchers, but also, of course, pharma and for other for-profit entities, citizen scientists. We think of this as a tool to engage educators, students, trainees along a continuum, and patients as they are health curious, and there are lots of stories of data-minded patients and patient groups who have explored things and discovered things that we want to engender. And so, I find that I'm ending a lot like many of the other folks up here in thinking about, how we want to share data across a number of different resources, and we need to have common semantics and syntax across how we do those. We are involved in GA4GH as one of the driving projects, and a number of these other large resources, as well as helping shape the EHR data standards that we're using in a group called Odyssey. And trying to map that as well. So hopefully that will map onto the clinical data that we have coming, and that explosion that we see, that we all agree is coming in the future. So thank you very much. I'll end with a picture of our teams.